In this video, I will show two plant species with distinct vegetative trait and relate this trait to genetic function we learn in this course. The first plant is a fern, belonging to the pteridophyte, which are vascular plant that reproduce through spore. The second is Melastoma malabatricum, commonly known as senduduk bulu, and angiosperm. Ferns exhibit several unique vegetative traits. First, their leaf develop through seasonate vernation, where young fronds are tightly coiled and gradually unrolled. This process is regulated by class 1 knot gene, which remain active in fern leaf primordia. Whereas in flowering plants, knot must be switched off. This activity allows fronds to branch repeatedly, forming structure like pinnae and pinnacles. Next, ferns fronds are megaphyte, large leaf with highly branched vernation. Their development depends on auxine guide patterning, mediated by pin-auxine efflux carrier, which generate auxine maxima to position pinnacles and pinnacles, giving the fronds its complex architecture. In angiosperm, auxine primarily regulate more intricate organogenesis, including veins, flowers, and fruits. Ferns also produce adventitious root from rhizome rather than developing the primary root system. Root identity is controlled by early diverging development regulator. Fern use CR-WOX-A for adventitious root formation, while angiosperm rely on WOX-5 for true root apical meristem. Finally, many ferns possess a siphonous style, a vascular arrangement with leaf gap. This pattern is guided by HD-ZIP3 homolog, while both ferns and angiosperm use HD-ZIP3 gene. Fern from sapolestali, where angiosperm develop compact vascular bundle capable of secondary growth.
The next plant is senduduk bulu, or the scientific name is Melastoma malabatricum. Senduduk or Melastoma malabatricum is an angiosperm, or in details, it is a flowering seed plant. It usually found in grassland and forest habitats. It also can be used as a medicine plant, which is it can treat a small wound on the skin by heal the blood vessel. As you can see in the picture, this plant has a melastoma berries that you can eat and pluck from the tree. First of all, the leaf arrangement of this plant is it has a large opposite or a walled leaves, or it is a reticulate vernation. And also, the gene link is Canops repression, PCP genes, and auxine maxima, or we call it as PIN1. Next is shoot branching of the Melastoma malabatricum or senduduk. It is an axillary branching and strong apical dominance. And the gene link is auxine transport genes, PIN1, cytokinin IPT, and strigolactone genes, or MAX or CCD. Next, let's move to the apical dominance. This plant has a strong apical dominance, and the gene link is AUX or IAA. ARF maintain apical dominance. Besides that, the root system of this plant is it has a true primary and secondary roots, and the gene link is WOX5 LRP genes and AUX or IAA, and also ARF7 or 19. Other than that, the vascular traits of this plant is it has a defined vascular bundles. Vascular bundles is a transport system of a vascular plant that transport water and also a nutrient. Last but not least, the growth habit of this plant is it is a shrub-like and erect plant. In conclusion, both plants show how the genes like KNOTS, PB1, PIN, and LBD control the vegetative traits such as leaves, growth, and roots. These differences of the genes contribute to the evolution of plant shapes that we can see until today. That's all from our group. Hope you enjoy watching our video. Till next time, tata!